molecule, adenine, a nitrogenous base, and a string of phosphate groups. Now let's construct an ATP molecule. Notice how the sugar ribose attaches to a nitrogenous base adenine, which is also bonded to a chain of 3 phosphate groups, the triphosphate group.
So I don't care that you know where these things are attached. I just really care that you know the 3 parts of it. And really importantly is that the 3 phosphate groups are the tri in ATP, the T in ATP.
During cellular respiration, glucose releases energy. This energy is stored when a phosphate group is added to ADP, adenosine diphosphate, forming ATP or adenosine triphosphate. ATP releases energy when the covalent bond between phosphate groups breaks during hydrolysis.
So when we say that ATP is an energy source, it's because in the process of turning ATP into ADP and breaking that bond, it's releasing all of this energy.
This energy is used to drive other biochemical reactions.
So the breaking of that bond that releases energy is that energy coupling. That energy can then be stored and used elsewhere. I don't remember what this is. This is an image of, it's just showing you when you add water, it's a hydration reaction. So you break it up, right? To convert ADP back into ATP, we would take water out. So it'd be a dehydration reaction. And now we're smashing that phosphate group back on there. So in the process of converting ATP into ADP by taking off that 3rd phosphate group, I would know that it's a hydration reaction, which means that you add water, and that in breaking that 3rd phosphate group off, we're releasing a ton of energy. So that brings us then to enzymes and understanding enzymes. I don't know if you guys have covered this yet in lab or if you've done any, however y'all are doing experiments this year, but an enzyme is basically a catalyst that helps speed up a reaction, but it's never used by the reaction. It's constantly recycled and can be used over and over and over again. And what an enzyme does, it basically is something that helps. So there needs to be in any kind of reaction that happens, even in an exergonic reaction where energy is given off, our products have less energy than our reactants because we got rid of that energy. Even in a reaction like that, you still have to add a little bit of energy. It has to use some energy before it can, get to the top of the hill and then roll down and give off all this other energy. So what an enzyme does is it doesn't make more products, and it oftentimes doesn't speed up the rate of the reaction, but it can. What it will do is it always lowers the initial energy that has to be added to a reaction to make it occur. And that's called the energy of activation, or E sub A right there. So when we don't add an enzyme to a reaction, then you're the black line, that is the input of energy is pretty high and then it falls off. When you add an enzyme, you lower the energy of activation, but you notice that you still have products. The amount of time it took you to get to those products, is pretty much about the same. It doesn't change how much energy is given off. It just lowers how much energy has to go into it initially. So enzymes lower the energy of activation. And enzymes, I would know the structure of what an enzyme and a substrate are. So an enzyme is usually a protein, and it's very small most of the time. And your substrate is the molecule that's going to react with the enzyme to spit out your reactants. So an enzyme comes along and it gets put inside of a substrate. And when the enzyme and the substrate have bound to one another, they're called an enzyme substrate complex. And then when the products are made, it'll spit that enzyme out and it comes back around to grab onto another substrate. So I want to go and show you here. Well, I guess not. Let's look at the key terms first. So the place where the enzyme attaches is called the active site. And a lot of times it's called it's an induced fit, which means that it fits, it's meant to be there. So there's all these enzymes in our body, and they're not just making reactions happen all the time. They are specific to a specific substrate. And when they go into that substrate, they'll change the shape of it, and the substrate they shake hands. He'll hug it and they'll hang on to one another. So our actually our enzyme is way bigger because it is a protein, sorry. So our little substrate is floating around, and it goes into the active site of our enzyme. And our enzyme, now that they're shaking hands, it'll a lot of times it's like a transformer. It'll change total shape so that it's hanging on to that substrate. The enzyme and the substrate are stuck together until they've made the products that they want to make. And then it lets that substrate out, usually lets the substrate out as the product. And the enzyme is still there, left behind to pick up another substrate later on. And so the substrate complex is that right-hand side where the enzyme's active site has caused an induced fit around the substrate and changed its shape. So there are a couple things that can affect an enzyme's activity. Temperature and pH are the big ones to really know. So there is an optimum temperature for everything to occur. And so if we used to always do a lab where we would perform different experiments at different temperatures. You'd add ice to something, you would heat something up with a Bunsen burner, you would change things to see how quickly your reaction happened. And every enzyme has a temperature where it works at its optimum level. And I don't really know what this, this is showing, it's just two totally different reactions. So humans, we like things a little bit cooler. A thermophile is something that loves to be in hot environments. So it would be in like, springs or something, hot springs, or in a volcano or something like that. But our body does its best work at this optimum temperature, 37 degrees Celsius, which I think is like 98 degrees Fahrenheit. I'm not really sure. Y'all can do that little calculation, but that's whatever it is. It's what our body temperature normally is. And so when you have a fever and your body's off, if your fever gets too high, your body will shut down and it will stop doing the work that it needs to do because it's outside of its optimal temperature range. Same thing if you get too cold, your body will stop working because it needs to be at that optimum temperature. And then pH is the same thing. So a stomach, and that your stomachs are full of acid. That's what breaks down your food. And so they really like a very low pH, a very acidic environment. And then our intestinal enzymes like it a little bit more neutral. So certain enzymes work better in certain pHs. So if your pH is off, if you're way too acidic in your stomach, then you start eating up everything. And if you're too basic in your stomach, then it's not going to break things down properly. And that's why you take medicine in the form of buffers to fix that kind of thing. So sometimes I want to see if I want to go here first before I say it, maybe not. So sometimes enzymes will have a second part to it, a non-protein molecule that helps the reaction happen a little bit quicker. And those are called cofactors. And then if it's organic, we call it a coenzyme. And so, I kind of just want to show you this competitive and non-competitive inhibitor. So if the purple thing is our enzyme, and then you have your substrate that's going to fit in there like, pegs in the kids game perfectly. Sometimes we get competitive inhibition. So you have a competitive inhibitor that can fit into part of that active site, but not exactly, and not enough to make the reaction that wants to happen. But what it does is it blocks it. So it blocks it and it keeps the substrate from being able to bind with the enzyme. A non-competitive inhibitor binds at another part of the enzyme, and it changes the active site so that it can no longer fit in there perfectly to make the reaction happen. So we can inhibit certain reactions by introducing some sort of an inhibitor. Competitive fits in the active site, and non-competitive will fit someplace else. Um, and then there's some types of regulation of our enzymes, allosteric regulation. So if you look at this teeny tiny little picture, our enzyme is the purple complex right there. And we have 4 active sites on this particular type of enzyme. And so they have an allosteric activator that will stabilize that form. So that little green candy corn looking thing will help keep that 4-part enzyme in its perfect shape so that all those substrates can fit in there perfectly. When you sometimes have an allosteric inhibitor, it'll fit inside there and keep it in a funny shape so that you can't. So it continues to inhibit it. So those are like examples of those coenzymes in whatever slide that was in those cofactors. One of those slides up there. Trying to think if there's much to say about this. Pretty much the same thing. So, when you have a substrate, there are times when your enzyme will be in an inactive form, and then that's usually when you have that allosteric regulation coming in and changing its form so that it can now accept the substrates. It's not being inhibited, it's just not in its proper form right now. And then a lot of times there's feedback inhibition. So a reaction happens, and when you have enough of an end product of something, it actually shuts down itself. So this is an image 619 kind of shows that it's not giving you any specific thing of what is well, I guess it is, but you've got your substrate that's binding with your enzyme, and when you have enough of this end product down here, it actually will fit the end product. The product will fit into that non-competitive active site. So it's the end product is essentially non-competitive inhibition. It's made enough of itself, it's saturated the place, and so it tells the enzyme, quit doing the reaction. We have enough of it. We don't need any more of whatever I am, and it will actually shut its own enzyme down. So that's feedback inhibition, and that's actually positive inhibition right there. Okay, I'm going to switch to chapter 7. Are you guys good on chapter 6? It's basically enzymes and how they work, lowers the energy of activation, the types of energy that there are, and then the 2 laws of thermodynamics, and then just understanding what free energy is, just kind of a lot of glossary terms. Okay, so I'm going to stop sharing that. And I'm going to share 7. In my opinion, at least when I was in school, I thought that cellular respiration to me was very confusing, because when I think of respiration, I always think of breathing. And this isn't necessarily your cells taking in oxygen and breathing. It is called respiration because oxygen is used, but don't think of it as anything with your lungs at all. This happens all throughout your body and not just in your lungs. So the 5 main things that I would say for cellular respiration to help you kind of narrow it down in what to know: understand what a redox reaction is, and then all the parts that have to do with the redox reaction. And then in cellular respiration, there are 3 stages. So you want to be able to name each stage, tell me what goes into each stage, what the reactant is, and then what the products are, and also where it occurs inside of the cell. So all of cellular respiration, or the organelle that's very important in cellular respiration, are mitochondria. And the general flow of energy in the universe is that all energy starts with light energy, and it's taken into plants through the process of photosynthesis, which is chapter 8. During photosynthesis, organic molecules like sugars, starches are made. We consume those things. And also, we know that plants give off oxygen, right? So the sugars that are made by the plants and the oxygen are used in cellular respiration. The energy organelle for our plants are chloroplasts. The energy organelle for humans or for animals, not just humans, for any non-plant, is the mitochondria. So most of cellular respiration is occurring in the mitochondria or very close to it. And in cellular respiration, our end products are carbon dioxide, which we know our plants need, and water, which our plants also need. And what else is made? So those are like trash products. We don't use those. What our plants use is the trash. But what we need is all of this ATP, all of this energy that we can now take to do cellular work. So cellular respiration is basically harvesting and producing tons of ATP so that the rest of our body can use that energy to do all the things that it needs to do. So your muscles can flex, so you can walk, so you can run, so you can do whatever, so your brain can work too. So when you break down those organic molecules, and remember our organic molecules were lipids, sugars, proteins, and nucleic acids. Well, we are consuming really proteins, lipids, and sugars. If you consume a very complex thing like lipids, it gets broken down until it's essentially just sugar. So when you eat very simple sugars, you get a quick burst of energy, and it will make more sense here in a minute. When you eat something much more complex, it allows you to have long-term slow energy being released. When you eat too much fat, it gets stored because it's so complex that you don't need all of it at that one time. So we're not going to look at what happens when you consume lipids or proteins. If you took like biochemistry, you would probably look at that kind of thing. We're just going to make it real simple and look at it with the simplest sugar that you can imagine. So when you break down those organic molecules, you give off energy in the form of ATP. There's different types of production of ATP. Fermentation is when we don't have oxygen. We'll talk about that in a minute. And that's usually called anaerobic respiration. Means nothing. Aerobic means oxygen is present. So there's no oxygen there. Aerobic respiration is not like aerobic exercise necessarily, but that is where oxygen is present. And so we're able to go through the whole 3-step process. So I would definitely know this general equation for what cellular respiration is. Cellular respiration includes aerobic and anaerobic parts to it, but it's mainly most of it is aerobic. So when we talk about cellular respiration, we're talking about when oxygen is present. When you're starved for oxygen, that's when fermentation occurs, and in our bodies it's usually in the form of like lactic acid builds up, and that's why you get sore in your muscles. So the C6H12O6 is your simple sugar. That's just one little tiny glucose molecule. Plus 6 oxygens are our 2 reactants. And then our products are CO2, H2O, and energy in the form of ATP and heat. So I would absolutely know what that equation is, because I can promise you're going to see that again on a quiz, in the homework, and definitely on a test. So part of cellular respiration is we will do a redox reaction. A redox reaction is when you transfer electrons between reactants. And it's confusing because it's kind of contradictory to the way that your brain wants to think about it. So when you're oxidized, you lose electrons. But if you think about it, an electron is a negative charge. So a reduction is when you gain an electron or you gain a negative charge. It is always so confusing for students to think something gets reduced if it gains something, but think of it in terms of electrons and charges as opposed to like objects that you get. So if you're oxidized, you become more positive. You give up your negative electrons. If you're reduced, then you gain the electrons, and now you're more negatively charged. And this is a generalized redox reaction. So if X has all the electrons, it's going to transfer it to Y. So Y becomes reduced because it now has a negative charge, and X becomes oxidized because it lost its electrons. It now gained a positive charge. And then if you looked at it with sodium chloride, sodium becomes oxidized because it's now positive, and chloride is negative because it gained that electron. So it's reduced. And this will make sense when we talk about the electron transport chain. That's why it's kind of stuck in here when it seems like it should have been in our chemistry chapter. So the other thing that is confusing is the person who gave up, or the molecule that gave up the electron, is the reducing agent, and the electron acceptor is the oxidizing agent. So if we go back up, I like the really simple one. So X became oxidized, but since X gave away its electron, so it is the reducing agent, although it became oxidized, and the electron acceptor, which is Y, became reduced, but it was the oxidizing agent. So the agent is the reactant, is on the reaction side, and what becomes reduced or oxidized is on the product side. You can get that turned around in your brain so many times. So you just kind of have to look at it and think about it, and look at a couple different examples of reactions in the book, and watch the animation from it and see if you can keep it straight in your head, because it is just the words oxidize, reduce, and what they gain and lose is, at least to me, it's very easy to get them confused. So during cellular respiration, the reason why we're talking about redox reactions is because in cellular respiration, glucose becomes oxidized and O2 is reduced because it turns into water. It gains those hydrogens, or those electrons that were on the hydrogen, that are on the hydrogen. So cellular respiration is a type of redox reaction. So if glucose becomes oxidized, then it is the reducing agent, right? And if oxygen becomes reduced, then it's the oxidizing agent. And then there's some other things that we're going to look at. So we talk about ATP as having a lot of energy, but there's another molecule that also is really important in storing some energy, and that's, you're going to see byproducts of NAD or NADH. And what's important about that is that NADH will have all these extra hydrogens, electrons stuck to them. So when you have an overload of all these hydrogens, we're able to get this little transport chain to get going. But we're kind of stepping ahead before we even talk about the electron transport chain. Oh, okay, there you go. So glucose becomes oxidized and turns into CO2. So it got rid of a ton of its hydrogens, right? And it gave them to the oxygen. Oxygen now becomes H2O. So CO2 was oxidized. The reducing agent was glucose. Oxygen becomes reduced to H2O because it gained negative electrons. And so the oxygen was the oxidizing agent.
Are the equations always set up like that, where the oxidized one is before the reduced one?
No. It just happens that all those examples were like that, but no, it doesn't matter. It doesn't matter where they are, at least on their respective sides of the arrow. So NADH has all those extra hydrogens. When it gets all these hydrogens, it's going to give those electrons. So I guess I had my slides kind of out of order there, but just remember that NADH is very important because it's harboring all of these hydrogens, these electrons, that it's going to give to the electron transport chain, which is how we get the majority of our ATP. So NADH is really important in generating tons of energy. Okay, so this is an overview of where everything happens in cellular respiration, and then kind of just some, it kind of shows you the byproducts of cellular respiration. So it is basically, it's three main parts: glycolysis. There's a little tiny, like, not true part. It's just like a prepping phase called pyruvate oxidation. Glycolysis occurs outside of the mitochondria. So it's in the inner cellular membrane, or the cytosol. It's inside of the membrane of the cell, but it's not inside the organelle mitochondria. When the glucose is converted into something called pyruvate. But pyruvate can't get through the mitochondria. And so it has to be converted into this thing called acetyl CoA. So it's not a true part of cellular respiration, it's just to get it into the mitochondria. The second or the middle stage of cellular respiration is the citric acid cycle. And then the part that makes the majority of energy is our electron transport chain. The citric acid cycle is often called substrate level phosphorylation. And then the electron transport chain is called oxidative phosphorylation. So let's look at those. We'll go through all of those steps. In your book, you're going to see images of like, all 12 steps of the citric acid cycle. You don't have to know that for biology. I want you to just really know what goes in, what comes out, and where it happens. I don't need to know every single step along the way. You will have to know that if you take biochemistry. You'll have to know every single thing and what the total chemical reaction is for every single simple step. But for this, we're just going to look really broad. What goes in, what comes out. So oxidative, that electron transport chain, the purple that is oxidative phosphorylation, it accounts for about 90% of the energy that's generated. So it's really, really super important. It gives us so much energy. But it can't happen all on its own. We have to get our glucose broken down into the proper thing to make that occur. And then in the citric acid cycle, we call it substrate level phosphorylation. Phosphorylation meaning that you are adding you're making a ton of ATP, and then that tri gets popped off and it makes all this energy that we talked about in chapter 6. So for each glucose that is broken down, that is oxidized into CO2 and water by respiration, we make about 32 molecules of ATP. So it doesn't seem like a lot, but that's why if you're eating very, very simple sugars, you're getting, bam, a big burst of energy, and then you crash because you have nothing left. Nothing continues to get broken down. So glycolysis is the only one within cellular respiration that will occur whether or not oxygen is present. But if oxygen is present, then we can move on and we can move into the mitochondria and go through cellular respiration. So glucose is often called our energy. There's different phases of glucose, sorry. So just like with anything, you have to, if you're starting a business, you have to invest money in order to start making money. Same thing with making energy. You have to add a little bit of energy in order to produce more energy. So in glucose, the first part of it, where we have an energy investment phase before we get the payoff. You have to invest 2 molecules of ATP in order to produce 4. But you're also producing a little bit of NADH, which is really, really important because we need that. We're going to need all those hydrogens a little bit later on. So although you produce 4 ATPs, you use 2. So I would remember that in glycolysis, the first part of cellular respiration, your end products are 2 pyruvate molecules, only 2 ATP, even though it made 4, and then 2 NADH. So that could very easily be a trick question if you don't read it properly. And I really do try not to write trick questions, or if I'm picking from a bank, choose trick questions. But how much total ATP is formed is 4. How much is gained, or what is your net, is only 2, because you had to input 2 in the beginning. And I wonder, I kind of want to show you. Okay, so just kind of remember in the back of your head what that term pyruvate is. You basically split glucose in half, and you made 2 things that we call pyruvate. Just remember that in your head, and then you'll see it in just a second. So if oxygen's present, then those pyruvate molecules, it was sugar and now it's 2, they will now get to go into the mitochondria, and they finish the rest of cellular respiration. CO2 is released, and the pyruvate is converted into acetyl CoA. This is going to make a whole lot more sense when I show you pictures of it. And then you produce another NADH. I kind of want to skip forward and show you the picture because I feel like that is easier to know. So, well, so you know what what your glucose molecule looks like when that thing it's a six carbon chain, right? It's C6H12O6. Well, now we break it into three little miniature tiny, they're not even sugar molecules anymore, but we split our sugar into our six ring sugar into three carbons. And now you have two. And in this, they're all in the cytosol as glucose and then pyruvate. Well, now they're small enough that they can enter the mitochondria. So when they enter the mitochondria, carbon dioxide is given off. And then we add something called CoA, which is up here. There, that coenzyme A, to make the pyruvate turn into something called acetyl-CoA. Acetyl-CoA is the only thing that can go into the citric acid cycle. So we just had to have an enzyme change it a little bit so that it can be added to the citric acid cycle. So sugar has to be broken down into pyruvate, and then it has to be turned into acetyl-CoA. So if you, well, I guess it kind of shows it. So so in the greenish bluish box right there, glucose for glycolysis is our reactant. Our product is pyruvate. But citric acid cycle doesn't take the direct product of glycolysis. It has to change it into that acetyl-CoA first. So if you were asked, and the citric acid cycle is just a cycle, it just cranks and it cranks and it cranks. And there, right there. So acetyl-CoA goes in, and then it binds with an end product, and it just keeps going and keeps going. And as it keeps continuing, the cycle goes, it kicks out more CO2. It makes a lot of NADH, and it also makes something else called FADH. All these things, it doesn't matter what they look like. What you want to know is that they have those hydrogens there, and those hydrogens are going to be stored and used for the electron transport chain, because hydrogens are an electron. So energy is produced, NADH and FADH2. And your end product is still reused over and over again as those glucose molecules get broken up. So if you're asked, if you consume if one sugar goes into cellular respiration, how many times does the citric acid cycle turn? And your answer should be 2, because that one sugar is broken into the 2 pyruvates, then converted into acetyl-CoA. If you're asked, if you have 10 sugars that go into cellular respiration, how many times does the citric acid cycle turn? Well, it's always times 2, because your sugars are doubled essentially when they get to the citric acid cycle portion. Okay, hopefully that made sense. Citric acid cycle has 8 steps. I don't care that you know what all those 8 steps are. If all you want to know is that little really simplified version of it, that's fine. Just know that it's just a little cycle that keeps turning and turning and turning. And every time an acetyl-CoA comes in, that gets that little wheel to turn one more time. But it's the NADH and the FADH2 produced by the cycle that are stored in the inside of the mitochondria that are so important. And that is what that ugly thing looks like. So glycolysis and the citric acid cycle only produce 4 ATP molecules, but there's all this potential energy in the NADH and FADH2 to help produce more ATP. And so that happens through the electron transport chain. And this is where water is formed because of all those oxygens that were reduced. ATP is not produced directly by a function of, okay. So I want to show you a picture first, and then I'll go back up here. So when cellular respiration has occurred, in the inner membrane space, this darker orange color is where the citric acid cycle was occurring, right? So we have all this extra storage of NADH and FADH2. Well, those things are going to pump. In the the orange part is outside of the mitochondria. The yellow is inside of the mitochondria where citric acid cycle took place. So this NADH and the FADH2 is going to lose its hydrogens, and they're all going to get shoved outside of the mitochondria. And when they get shoved outside the mitochondria, they accumulate, and there's this huge number of electrons sitting out there that don't really want to be there. You know, think of diffusion, right? It's too complicated and complex, and there's way too many of them there. So they accumulate, they become very, very condensed, and they want to get across the membrane and get back in to even things out. In that process of all these hydrogen molecules accumulating and then being allowed to move back in, you get this crank turning. And every time a hydrogen pops through, it turns this crank. And every time it cranks, it gives off all of this ATP. It gives off ATP. And so the NADH and the FADH2 having all that potential energy because they have the hydrogens to go across the mitochondrial membrane and then get shoved back through because there's way too many out there. They want to go back inside. That produces all this ATP. So remember how the mitochondria has those has so many curves, and you know it moves and it bends in the in its own membrane, in the mitochondrial membrane. That allows so much more surface area for ATP to occur because it has more spaces now for that little crank to be turning. There's more places for the electron transport chain to be embedded inside of the membrane. So now I'm going to go back up because I feel like looking at that makes it a little bit easier to understand. This is just a breakdown of it showing that NADH and FADH2 give up their hydrogens and get shoved across the membrane. So energy is released by electron transport when the hydrogen gets pumped back into the inner membrane space. And yeah, okay. So you can think of it, well, that's just another version of the way it looks. If there is an animation of what it looks like three-dimensionally, like an electron microscope version of it, and it really does kind of look like that, where it looks like it's just like a little crank that kind of turns and turns and turns. So yeah, that's the same picture. Okay, so our overall yields from cellular respiration. During glycolysis, no oxygen is needed. Glucose goes in, 2 pyruvates come out. We had to add a little bit of energy, so we only get 2 ATPs from it, and that's through something called substrate level phosphorylation. This happens outside of the mitochondria. It's the only part of cellular respiration that's not inside the mitochondria. Glycolysis, 2 pyruvates are formed. They have to be converted into acetyl-CoA. So we add that CoA enzyme or coenzyme, and that changes its shape so it can now go into the mitochondria, and it can now be added to the citric acid cycle. Citric acid cycle gives us very little energy also, just 2 ATPs, but tons of the NADHs and the FADH2. So we have lots of potential energy. So we're able to form this disconnect. There's too much hydrogens on one side, and it wants to shove them back across the membrane. And that gets the crank turning and tons of ATP to be formed. So you think, well, why don't we just do electron transport chain? Well, you can't do it unless you have all of those hydrogens. So all of these previous steps are very important to getting all the stuff that you need for making all that energy, for making all that ATP. So if you don't have oxygen present, then you go through fermentation or anaerobic respiration, which is no oxygen is present. All of the processes, whatever kind of anaerobic respiration you have, or if it's aerobic, it always starts with glycolysis. So, okay, so fermentation doesn't use the electron transport chain. So that's why we need oxygen to survive, because while our muscles can do a little bit without oxygen, it can't do enough. We cannot get enough energy from it because we never get to electron transport chain. So there's different types of fermentation. There's different types of anaerobic respiration. fermentation is one, and then lactation or lactic acid fermentation is the other one. So alcohol fermentation converts that pyruvate. So glycolysis happened, glucose got turned into pyruvate, and then we turn that pyruvate into ethyl alcohol. So when you ferment something, that's how they make yeast for brewing, winemaking. We use yeast for baking because it also gives off a lot of oxygen, and so it makes things your breads and things very fluffy. So alcohol fermentation, you produce ethanol, and then so in this case, you make a little bit of ATP. NADH is constantly regenerated over and over again to be used in that glycolysis. But alcohol fermentation, you start with glucose, you end with ethanol. And then lactic acid fermentation is converted into lactate. Lactic acid is what builds up when you work out really hard and your muscles become starved for oxygen. So if you're running and you're sprinting and you start to feel the burn, it's because you have used up all the oxygen in your body, and so your muscles are starved for that. And you actually have little tears in your muscle, and the lactic acid builds up in those tears, and that's what sore muscles feel like. So the more sore you are, the more lactic acid you have in little teeny tiny tears. And the idea is that as those little tears repair themselves, you grow a little bit more muscle. So lactic acid fermentation is when glycolysis occurs, the sugar turns into pyruvate, just like with everything else, and that is turned into lactate. So I would just know kind of the byproducts or the end products of the different types of fermentation. I would know that glycolysis still happens, glucose still comes in, or just what comes out is a little different. And that we have lactic acid fermentation would be like with mussels most of the time. And then alcohol fermentation is making ethanol, makes alcohol, yeast, that kind of thing. Actually, we starve the yeast for oxygen, and then that's how it makes the ethyl alcohol. And there are some animals that always have to go through fermentation or anaerobic respiration in order to survive. They cannot survive without the presence of oxygen. These guys are called obligate anaerobes. They do not, if they're in anywhere with oxygen, they die. And then there are some things that are facultative anaerobes, and that means that they can live with oxygen or they can live without it. So they're very well adapted to environments that will lack oxygen at periods of time, but will not die if oxygen is then present. I think that's the last slide of that, which is perfect timing. Do you guys have any questions? I know cellular respiration is tough, so it's going to take watching those videos and reading it and doing a lot of studying on your own. I never learned it myself, just sitting in a lecture hall, so it definitely has to kind of be self-taught. But I've hopefully tried to simplify it. I don't care that you know all the parts. I really just care that you know what goes in and what comes out. Do y'all have any questions about 6 or 7 for me?